All right, welcome to Standing for Truth, everybody. I am your host, Donnie, and today we have an extremely exciting program that will focus on junk DNA. We are going to be answering many very, very important questions about junk DNA and how exactly it relates to creation and evolution. Looks like we've already got a great chat. Thanks, everybody, for showing up. And Dr. Fuz Rana, thank you so much for giving us your time for this important presentation. I really appreciate it.
Well, Donnie, thanks for having me. It's always a lot of fun to talk about these kinds of issues.
Amen. Amen. Well said. It's certainly a privilege, Dr. Rana. So a few of the questions we're going to be addressing today is junk DNA still a valid argument in favor of evolution? Does most of the genomes of living organisms, including humans, consist of evolutionary leftovers, genomic fossils, and genetic baggage? Why would God create genomes of junk? Wouldn't a designer create genomes of treasure and function? These questions and more will be answered today. Now, before we get right into your presentation, Dr. Rana, since it's your first time on the show, I do want to give the audience a brief introduction into who you are and how they can find more about you, because I've spent hours now reading through your articles and I highly recommend them. So Biochemist Fazale, or Fuz Rana, is VP of Research and Apologetics at Reasons to Believe, an organization dedicated to communicating the powerful scientific case for God's existence and the Bible's reliability to Christians and non-Christians alike. Fuz earned a BS in chemistry with highest honors, West Virginia State College, and a PhD in chemistry with an emphasis in biochemistry from Ohio University. He pursued postdoctoral studies on cell membranes at the universities of Virginia and Georgia, and worked as a senior scientist in research and development at Procter & Gamble. Fuz has been published in peer-reviewed scientific journals such as Biochemistry, Applied Spectroscopy, FEBS Letters, Journal of Microbiological Methods, and Journal of Chemical Education. He also holds 2 patents, authored a chapter on molecular convergence and intelligent design for the nature of nature, and co-wrote a chapter on antimicrobial peptides for biological and synthetic membranes. In addition, he has delivered numerous presentations at international scientific meetings. In other words, Dr. Rana, you're more than qualified to be discussing these topics. I appreciate it. Is there anything else you kind of wanted to add there? Anything that I may have missed, Dr. Rana?
No, that was a very generous introduction, so nothing to add.
Okay, awesome. I appreciate it. Check the description box, everybody, for more in terms of Dr. Rana's work and articles he's putting out. So that being said, I'm going to hand it over to you, Dr. Rana, to get right into the topic and the presentation for this evening.
Yeah, and I'll just do, I'm hoping about 30 minutes or so of a presentation, and it's not going to be complete by any means, but hopefully we'll fill in the gaps during the discussion in the Q&A. And I'm going to go ahead and share my screen to get us started. And I'm not quite sure how to do this any more eloquently than I am right now. So here I am bumbling around, but all righty, here we go.
We got it. Looks good.
Okay, thank you. Yeah, and what I thought I would do in terms of addressing the issue related to, quote unquote, junk DNA is to take a look at the ENCODE project, because this is a body of work that I think has really important bearing on the question of the structure and the function of not only the human genome, but really by extension, the genomes of other organisms. And, I don't know if anybody remembers, I think it was June of 2001, or no, June of 2000, if I'm not mistaken, how the time flies, when Bill Clinton, then President Bill Clinton, hosted a press conference where you have Francis Collins, who headed up the Human Genome Project, sponsored by NIH, and Craig Venter, who was the head of Celera Genomics, a private company competing with the public project to essentially sequence the human genome, where they realized that whoever got across the finish line first had a lot to gain by that accomplishment, where Venter, through Celera Genomics, is really trying to commercialize the information that was found in the human genome. And they ended up agreeing to lock arms and to call a truce to their competition, share information with each other, and collaboratively completed the first draft of the human genome sequence. And at that time, then President Bill Clinton had these remarks where he said, "Today we're learning the language in which God created life. We are gaining ever more awe for the complexity and the beauty and the wonder of God's most divine and sacred gift. With this profound new knowledge, humankind is on the verge of gaining immense new power to heal. Genomic science will have a real impact on all of our lives, and even more on the lives of our children. It will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases." And of course, the human genome is about 3.2 billion genetic letters in size. It's a massive amount of information. And this is a picture of me a few years ago at the Wellcome Trust Museum in London, England. And in the Wellcome Trust Museum, they have a display of the human genome. And they have these various volumes that essentially are listing out the human genome sequences, and the volumes are numbered according to the chromosome number for each of the sequences. And you can see how much information is harbored in the human genome when you look at 3.2 billion genetic letters. And of course, we have all of this information, but the question then becomes, what does that information actually mean? All right, how do we really make sense of this information? And along those lines, some of the very first work was to try to identify the number of genes in the human genome. And to everybody's surprise, there's only about 20,000 genes in the human genome. There are techniques you can use where you can kind of identify certain sequence markers that indicate the beginning of a gene sequence. And using that technique, estimates are there's about 20,000 genes in the human genome. And this is a far smaller number than anybody would have anticipated, which indicates that a vast proportion of the human genome is consisting of sequences that are non-coding, that don't directly code for protein products or for RNA products like ribosomal RNA and transfer RNAs and things like that. And so this insight from the human genome sequence at that time fit perfectly within the confines of evolutionary biology, where people argued that most likely 90 to 95% of the human genome consists of non-functional or, in colloquial terms, junk DNA, and that this finds explanation in something known as the C-value paradox. And the C-value is a parameter that refers to the amount of DNA in the nucleus of an organism's gametes, measured in picograms. And you could think of the C-value as roughly equivalent to the size of the genome. And it turns out that when people began measuring the C-value, they expected initially to see some kind of correlation between genome size and the complexity of organisms. And in fact, they saw nothing of the sort, but rather didn't appear to be any kind of relationship whatsoever between C-value and, again, organismal complexity, where some relatively simple organisms had very large genomes compared to much more complex organisms. And the ultimate resolution of this was to argue that most of the genomes of organisms are actually consisting of non-functional or junk DNA that was accrued in the genomes of organisms through a variety of different mechanisms, and that these junk DNA sequences actually are highly informative because they can give us a picture of the evolutionary history of organisms, that these are vestiges of an evolutionary history, and that the relationship or the lack of relationship, again, between C-value and complexity is really a reflection of the junk DNA content. And when it comes to the human genome, again, it looks like somewhere between 90 to 95% of the human genome consists of what appear to be non-functional DNA sequences, where a significant portion of the genome consists of what are known as retrotransposons, line and sign and long terminal repeat sequence elements. There's a very small portion of the genome that consists of exons, which would be the coding region. A significant portion are introns, and then there's some satellite DNA sequences, particularly associated with centromeres and with telomeres. But again, it looks like, again, a significant portion of the human genome is indeed non-functional. And in fact, we've identified the different classes of these so-called non-functional sequences. And with each class, we've identified what we believe to be mechanisms that could account for the origin of these different types of sequence elements into the genomes of organisms. And so for many people, junk DNA really represents one of the most compelling reasons to think that human beings must have an evolutionary history, and that evolutionary history is shared with the great apes and in other organisms. Because again, people are arguing that these sequences are the vestiges of evolutionary history. And of course, this begs the question, why would a creator create genomes of organisms where a vast proportion of the genome is non-functional? But more critically, why would a creator create genomes of organisms where not only are there a large proportion of non-functional sequences, but those non-functional sequences are shared with other organisms that would naturally group with, in this case, with human beings, like the great apes? Why would these sequences have such a high degree of similarity? Why would these sequences occur in corresponding regions in the genomes? Why do these sequences display a nested hierarchy where you could actually organize these sequences into groups of groups that presumably reflect an evolutionary history? And then also, why would these sequences, in the case of endogenous retroviruses or the retrotransposons, why would they bear similarity to viral sequences, viral genetic information? Or in the case of pseudogenes, why would these pseudogenes display similarity to quote unquote functional or healthy genes? And so these are really questions that are profoundly challenging to intelligent design, to some kind of creation perspective. And it makes sense, particularly if you are embracing a materialistic view of origins, to look at the patterns that we see with junk DNA as being compelling evidence for common descent. And so if we're going to respond to the junk DNA challenge and present a creation model, if you will, or a design model for genomes, we've got to address all of these challenges. We have to be able to show that these sequences actually are functional, that there's a reason why these sequences would be shared, that there's a reason why they would be organized as nested hierarchies, and that they bear similarity to, again, things like to retroviral sequences or to healthy genes. So this is really the task that's in front of us if we are going to make a case that the human genome reflects the handiwork of a creator. And there's a lot of different ways to unpack this challenge and to marshal a response to the junk DNA challenge to the design argument. But what I'm going to do in the presentation is really focus on the functionality of DNA and, of non-coding, so-called junk DNA. And, what I'll then do is bring in some comments about these other factors as we go along the way, but we can leave some of the details to our time of discussion and time of Q&A. So I understand that the presentation I'm going to provide here is not complete. It's only really a partial response. Now, the ENCODE project, you could think of this as being kind of like a quest for the Rosetta Stone for the human genome. And this project was launched shortly after the completion of the human genome sequence, or at least the first draft, where the goal of the ENCODE project was to try to catalog all the different functional elements in the human genome. And so ENCODE stands for the Encyclopedia of DNA Elements. And this project began as a phase 1 project, which was a pilot level study that looked at a small proportion of the human genome. And the initial results were rather surprising because it looked as if a significant portion of a small percentage of the human genome actually consisted of sequences that were serving some kind of functional role. And the assays that were used in phase 1 included measurement of transcription, with the idea that if sequences are being transcribed, they most likely are functional. The binding of transcription factors, the binding of histone, the modified histone binding. This is related to epigenetics. Methylation, again, related to epigenetics, where again, the idea is that these interactions are serving a gene regulatory role. And then also assays that looked at three-dimensional interactions with respect to components in the human genome. And in, I think it was 2011, phase 2 of the project was published, where a large number of different cell types had their genomes analyzed for functional DNA. There's probably about 200, 210 different cell types in the human body. I think in phase 2, somewhere around 140 to 150 of these cell types were evaluated. And what was shocking to everybody is it looked as if 80% of the human genome displayed some type of biochemical function that was interpreted as having actual function, that a significant portion of the human genome was functional. And at that time, Ewan Birney, who was heading up the ENCODE project, was interviewed by Ed Young for Discover, and he said, what's in the remaining 20%? Possibly not junk either. It's likely that the 80% will go to 100%. And we now have seen phase 3 and phase 4 completed. And while I don't know what the percent is for functionality, it really does look like as ENCODE project workers continue to fill in the missing gaps, looking at functionality in other cell types, as well as functionality at different times during the course of development and during the course of the cell cycle, that at some point, most of the DNA in the human genome appears to play some kind of functional role. The phase 2 project indicated that there were 4 million regulatory regions in the human genome, and these included 70,000 promoter regions and 400,000 enhancer regions. So that the number of regulatory elements in the human genome was far greater. And the implication here is that many of these regulatory elements are actually residing in portions of the genome thought to be junk, like long terminal repeats or the line and the sign sequences. In addition to the ENCODE project, there have been other studies that have been done that have discovered in a piecemeal manner functionality for different types and different classes of junk DNA. So for example, about 2% of the human genome consists of pseudogenes, which were believed to be the remnants of once functional genes. And there's different types of pseudogenes, unitary pseudogenes, duplicated pseudogenes, and then of course, process pseudogenes. And it turns out that these pseudogenes actually seem to be playing a critical role in gene regulation through what has been dubbed a decoy mechanism. In fact, recently there's been an idea that's been advanced called the competitive endogenous RNA hypothesis, which actually provides a really elegant framework for understanding not only the function of pseudogenes, but why pseudogenes bear similarity to their quote unquote functional or maybe their coding counterparts. And it has to do with how the decoy mechanism works. But this competitive endogenous RNA hypothesis explains functionality for not only duplicated pseudogenes, but unitary pseudogenes as well. And we're also discovering that some pseudogenes actually code for functional proteins. Now, one of the criticisms that is leveled against arguing that pseudogenes are functional is that, well, some of the pseudogenes are transcribed, and therefore, you could argue that they are functional, but a number of pseudogenes actually are not transcribed. And it turns out that there may actually be functional roles that these untranscribed pseudogenes are playing as well. But part of the challenge here is methodological, is that many times people are assuming regions or assuming pseudogene sequences are not being transcribed when in fact they are. And the challenge is it's very difficult to distinguish between a pseudogene and a coding gene sequence through some of the assays that are used to measure transcription. Line DNAs have been observed to play a role in X chromosome inactivation, as well as play a role in what's called monoallelic gene expression. Sign DNA is expressed during the cell's stress response, and it looks as if these sequences are playing a protective role as well as a role in gene regulation. Endogenous retroviruses look as if they actually play an antiviral role, and the way in which they exert their antiviral function is actually multivaried, that there's a number of different mechanisms, but all of these mechanisms actually are predicated on the similarity between these endogenous retroviral sequences and the genetic sequences of both retroviruses as well as other types of viruses. Elements of endogenous retroviruses like the long terminal repeats also play a role in gene regulation. Just recently, a research team indicated that these endogenous retroviral sequences actually play an anti-cancer role. That P53, the guardian of the genome, actually, when cells are stressed, which would take place when cells become tumorigenic, the P53 actually triggers the expression of endogenous retroviruses, which triggers essentially a response on the part of the immune system to clear those tumor cells from our bodies if we have cancer. So the bottom line here is that we're discovering that junk DNA is anything but junk, that it's actually functional DNA. And in terms of these shared sequences that we see, you could actually argue that the shared sequences are reflecting common design as opposed to common descent. And that we could also explain nested hierarchies through a common design model, if you will, a shared design model. And we'll get into the details of that if people are curious. I'm going to go ahead and skip ahead here and just close out the presentation by saying that I think it's legitimate to say that there's a new view of the genome that is emerging, that we really are in our infancy when it comes to understanding the human genome as well as the genomes of other organisms, and that it's very easy in the absence of having this understanding to draw conclusions about the genome that seem to indicate that most of the genome is non-functional. But as we're continuing to learn more about the genome, we realize that much of the non-coding regions of the genome that were dubbed to be non-functional or junk actually are serving very important roles in terms of regulating the expression of the coding sequences. Eric Green, who was the director of the National Human Genome Research Institute, when phase 2 of the ENCODE project was published, said, during the early debates about the human genome project, researchers had predicted that only a few percent of the human genome sequence encoded proteins, the workhorses of the cell, the rest was junk. We now know this conclusion was wrong. ENCODE has revealed that most of the human genome is involved in the complex molecular choreography required for converting genetic information into living cells and tissues. Okay, so I'm going to go ahead and stop there, and that hopefully just sets the stage. We can go ahead and just open it up for discussion and for a time of Q&A.
Awesome. Dr. Rana, that was a fantastic presentation, and so much good information. I know it's hard to just condense into a 30-minute presentation. That's why I want people to definitely check out the links in the description box for more on this and more on this in incredible detail. And Dr. Rana, you asked and answered all the right questions. Why would pseudogenes display similarity to functional or healthy genes? And why do we see nested hierarchical patterns in terms of these pseudogenes, junk DNA, and endogenous retroviruses? Is this a reflection of descent or design? I think you did a great job demonstrating why this can certainly demonstrate design due to the function that we're finding. So my first question, we got a ton of questions from the audience as well, Dr. Rana, but my first question after watching your presentation has to do with the ENCODE results. And we know that there's been a lot of evolutionists who have kind of been anti-ENCODE and have presented some challenges. What are your thoughts on that? And then how do we best address those challenges?
Yeah, and it's interesting because shortly after the ENCODE project published the phase 2 results, it initiated a significant backlash on the part of many evolutionary biologists who really decried the results of phase 2, where they argued that this would undermine the C-value paradox, that key tenets of evolutionary theory would be undermined if this indeed was correct. And the argument was that the ENCODE project scientists didn't really understand evolutionary biology to make these kind of outlandish statements. And so it ignited what you might call the junk DNA wars. And I think Carl Zimmer a few years ago wrote a really nice article, I believe it was for the New York Times, where he described the whole junk DNA wars, the junk DNA wars that was happening, not between ID proponents and evolutionary biologists, but within within the scientific community itself, where scientists who work in genomics found the results of the ENCODE project to be very helpful, whereas the evolutionary biologists again decried, the ENCODE results. And I a number of years ago did a debate at North Dakota State University on Darwin Day, where I engaged P.Z. Myers, and I think some of you may be familiar with P.Z. Myers. He has an award-winning science blog called Phalangula, and is an outspoken atheist as well. He's at the University of Minnesota Morris. And during our discussion time, a question about the ENCODE project came up, and what P.Z. Myers said was that the results of the ENCODE project most definitely can't be valid, because if they were, then it means evolution or key aspects of evolutionary theory simply can't be correct. And so it was interesting to me, because at that point I realized that what was happening was that the data was being evaluated in light of the theory, as opposed to a theory being evaluated in light of the data. And so it was almost like a bizarro world approach to science. And that attitude I see among many of the chief critics, of the ENCODE project results, is they really see the the results as being flawed because of the implications for really for the evolutionary framework. But there's been a number of of specific challenges. We can kind of get into some of those if you would like. But that essentially kind of sets the tone, I think, for what you're looking at with respect to the junk DNA wars is really two very fundamentally different views of the human genome that are at odds with each other, even within the scientific community.
Right. That's a great, those are some great points. And Dr. Rana, in terms of challenges, because, and correct me if I'm wrong, what we discovered, or one of the things we discovered with the ENCODE project, is that most of the genome is transcribed in RNA. I think it's about 80%. And the question becomes, if 80% of the DNA is transcribed, is this activity useless activity or beneficial? And I've heard a lot of evolutionists say, well, a lot of this activity is just spurious, or it's not really doing anything. It's just easier for the cell to just transcribe it because it would take more work to not transcribe it in terms of the cell. And so they'll say that this low-level transcription can actually be beneficial for the cell, even though it's not really performing anything beneficial or functional. What are your thoughts on? Have you heard that response before?
Yeah, and that's a specific example of maybe one of the most, I think, important critiques of the ENCODE project, and it's a critique that I think needs to be taken seriously, and that is, if we're measuring biochemical activity, does that activity actually translate into function? And I've seen this primarily played out with respect to transcription, as you're pointing out, as well as to the binding of transcription factors to the genome, where people are arguing that much of the binding is just simply random binding at non-functional sites. And to me, as a biochemist, when I hear people argue that a vast proportion of the transcription that takes place is essentially spurious, as you said, or just biochemical noise, that to me doesn't sit very well, because to transcribe a portion of the genome is a very energy intensive process. You basically, every time you're adding a nucleotide to a growing RNA chain, you're consuming 14 kilocalories per mole of energy. That's a lot of energy that's being released as heat for no purpose whatsoever, just to create transcripts. Not to mention that these transcripts would essentially clutter up the interior of the cell if they're not playing a functional role. So right away, that doesn't intuitively make sense to me. But as people have actually cataloged the different types of transcripts that are produced, it's very easy to show that these transcripts are actually playing a functional role. And so, for example, 2% of the transcripts are for protein, are protein coding. 2% are pseudogenes, which are playing a regulatory role. But there's a number of transcripts that are being transcribed, even though they're not translated, but they're playing a regulatory role. And these would include things like long non-coding RNA molecules and in small interfering RNA molecules and in microRNAs and things like that. And we know that these are again all playing regulatory roles as people pursue, the functionality of these transcripts. It appears as if they're playing regulatory roles. So it doesn't look like, with regard to transcription, that this is actually, a valid critique.
That's a great response, Dr. Rana. To me, it would be, as you pointed out, it would be a waste of energy and resources for the cell to transcribe all that information, which really just turns out to be spurious and useless. And the question that comes to my mind is, if this really were the case, wouldn't natural selection over millions of years of evolutionary history have removed all of this junk or all of this information if it really was just spurious?
Yeah, well, because that's a very good point, Donnie, because there are mechanisms that will suppress transcription. And so you would think that if you were really transcribing that proportion of the human genome or the genome of another organism. And it's biochemical noise, that there would be, evolutionary processes in play that would essentially suppress that transcription, that we would expect transcripts that are playing no role at all to be rather rare, not to be occurring at that high of a level.
Right, great point. And you mentioned P.Z. Myers, and I've read in a book, I believe it was by Dr. Lee Spetner, but he pointed out that Richard Dawkins, and I've actually got the quotes in front of me, in 2009, he said, "It is a remarkable fact that the greater part, 95% in the case of humans, of the genome might as well not even be there for all the difference it makes. But after the ENCODE result and a lot of the results have come about that you've been pointing to," he then stated, years later, I believe in a debate with Jonathan Sachs, he said, "I know there are some creationists who have jumped on it because they think it is awkward for Darwinism. Quite the contrary, of course. It is exactly what a Darwinist would hope for, to find usefulness in a living world." So to me, it kind of seems like they want the best of both worlds. If it's junk, it's evidence for evolution. If it's not, it's what they hope for, judging by Dr. Dawkins' quote.
Yeah, it's interesting because I've been at this for a while now, and I can remember even before I started working at Reasons to Believe, which was in 1999, that the junk DNA challenge was present and was raised against the design argument, and I didn't see anybody at that time making any kind of predictions from an evolutionary perspective that junk DNA was functional. In fact, it was the opposite. And very few people actually even bothered spending time studying, junk DNA sequences. A few people might have cataloged them and maybe did some work in terms of trying to understand how these sequences may have originated, or they may have used them as tools for phylogenetic analysis, but nobody really ever even thought to probe them for functionality. In fact, Craig Venter, who we saw a picture of earlier, was at one time part of the Human Genome Project. And he actually argued that it was a misguided approach to sequence the human genome because most of it's non-functional. What we ought to be doing is developing assays that measure transcription in the human genome and then characterize the transcripts, because only those sequences that are transcribed are going to be functional, and that we ought to build up our knowledge of functionality based on transcription. And in fact, published a landmark paper where he looked at transcription in neural tissue and discovered a huge number of unknown genes through that process. And was so frustrated by the approach taken by the Human Genome Project that he actually left and began pursuing his own research efforts along those lines in the private sector. But all that to say is that, people, when the Human Genome Project was underway and when the first draft was completed, nobody was making predictions about function. And in fact, if you look at anybody that was making predictions about function, it would have been people that held to an ID or to a creation, model approach. In 2000, I wrote an article where I put in print that we would expect the different classes of so-called junk DNA to one day turn out to be functional. And so it's only later on, when function is being discovered, that I'm now seeing, evolutionary biologists say, well, evolutionary theory predicted this all the time. And I think it's okay to say, look, there's, neo-functionalization, I think is not unreasonable, where the argument would be, hey, all of this so-called non-functional DNA is raw material that evolution can co-opt and modify and make use of to create new functionality. Okay, that's not an unreasonable argument. And neo-functionalization it seems to be a reasonable explanation when you're talking about isolated examples of functionality. But when you're talking about this wide scale level of functionality, that's where I think neo-functionalization is really problematic. It's one thing to say, hey, a handful of pseudogene sequences turn out to play some kind of regulatory role, and that this could be explained by neofunctionalization. But to say that the totality of pseudogenes is actually playing an integral role in gene regulation, to me, really makes it hard to argue for neofunctionalization at that point, just because of the nature of the evolutionary mechanism, which is unguided and undirected and historically contingent. So if neofunctionalization is your explanation, that works well when a fraction of the sequences are functional, not when the totality or the near totality of those sequences is functional.
Well, so many great points there, Dr. Ron, and I'm glad that you addressed the argument pertaining to neofunctionalization. And going back to a few things you said, which I completely agree with when it comes to predictions, testable predictions. As you've pointed out, it's the intelligent design advocates that can look to a lot of these results, for example, from the ENCODE project, suggesting so much genome activity. Intelligent design advocates like yourself can make future predictions on function pertaining to pseudogenes or retroviral-like elements, including ALU sequences. But from my understanding, it's the evolutionary community that are more so silent in terms of making predictions on function.
Yes, yeah, and again, it's only recently where you begin to see, people saying all along we thought this was functional, right? And and of course, I mean, again, to be completely fair, everybody thought that a portion of the non-coding DNA was functional. Right. These would be the regulatory elements and things like that. So, everybody agreed that there was going to be a portion that was non-functional. But I think the shock is the level of regulation that the non-coding DNA or the so-called junk DNA is actually playing or contributing. That to me is I think what is shocking. And in terms of predictions, in 2004, 2005, when we published the first edition of Who Was Adam, we addressed the junk DNA issue. And again, there was very little that we could really say at that time, about junk DNA. But along the lines of endogenous retroviruses, I went back just recently and was rereading that section of the book, and I actually made a prediction there about endogenous retroviruses not only being functional, but actually playing an antiviral role, serving as part of the innate immune system. And it turns out that in recent years, there's just a flurry of papers that are now being published showing that endogenous retroviruses, as well as endogenous viral elements, EVEs and ERVs, are playing a very important role as part of the innate immune system, is warding off viral infections. And so, again, that's another example of a prediction that's in print that is anticipating discoveries, and those predictions are flowing out of a commitment to a design, / creation model approach.
Amen. I think you nailed it there. Is the starting position is intelligent design and a creation, and these predictions naturally flow from that starting point. I find it interesting that I've heard a lot of anti-intelligent design people point out that intelligent design or the creation model doesn't make future testable predictions, and we know that would be the gold standard of science. But as you're pointing out here, no, it does. When we start from that position, we can make testable predictions that turn out to be successful.
Yes, and yeah, I want to be very careful as I say this, but oftentimes you hear this challenge leveled against intelligent design, against creationism, that if we embrace this idea that God did it, that it just would bring science to a screeching halt, that it would stultify science. But the fact of the matter is, in my lifetime, I've seen, the evolutionary framework actually, in the case of identifying function for junk DNA, really stultify science. Because many people, again, I gave you the example of Craig Venter, and he wasn't unique in his views. Many people just didn't bother to study, you know, the so-called junk DNA components of the genome. Why would you? If it's non-functional, why waste any time in resource dollars or research dollars, that are so precious to study, something that you don't think is functional, right? And it was really not. It was really, in the context of looking at, other questions that people have stumbled upon function initially for junk DNA. Now, I think you can find a lot of people that are studying junk DNA with an eye towards anticipating function, but this is a relatively recent change in mindset. And so you could argue that this evolutionary view of the genome actually stultified scientific advance, it delayed scientific advance.
If you anticipate treasure, then you're going to go into these studies looking for essentially treasure and function. And one thing I wanted to point out that you said that was perfect was because I constantly hear the critics say that we are misrepresenting them because they never not predicted everything would be junk. They predicted function. But I think you nailed it perfectly when you said, sure, they predicted some function, but not the level of function that we are discovering in terms of the genome. And I think that's the perfect response to these critics that put forth that argument.
And let me say this, I've never seen anybody produce a citation where there is that prediction from an evolutionary biologist that, that junk DNA would be functional. I'm not saying that people didn't make that prediction. I've yet to see that I cannot find that those kind of predictions. Now, if somebody can provide me with the references, I will be happy to stand corrected on that.
I've looked for that too, and I've not been able to find it. Not that it doesn't exist, I just haven't been able to find it. I definitely want to get this question in here. This is one of my own questions, and we got a ton of questions from the audience as well. Because it's just such an interesting topic. So you said, Dr. Rana, in your article titled "Functional Pseudogenes Are Everywhere," and I highly recommend people check that out. So you said proponents of the evolutionary paradigm offer this rejoinder. Yes, some pseudogenes are functional, but the vast majority lacks function. The vast majority is junk. The question is this true? Is pseudogene functionality actually rare and thus still support for common descent?
Yeah, and what we're discovering, again, with the competitive endogenous RNA hypothesis, is that unitary pseudogenes and duplicated pseudogenes are playing a role in gene regulation, where what we're learning is that genes produce transcripts, and those transcripts actually form kind of a family of transcripts that all are regulated by the same, biochemical machinery. There are these enzymes that will essentially, break down RNA as soon as it's produced. We used to think that gene regulation was only at the point where messenger RNA was produced, that you either allowed messenger RNA to be produced, or you shut it, or you inhibited its production, and that's how you would regulate gene expression. And there might be some translational control as well. But what we're now discovering is that there are these enzyme complexes that will break down RNA, and that this is setting up what biochemists call a feudal cycle, where messenger RNA is transcribed and it's broken down and it's transcribed. So in order to elevate the expression of a gene, not only do you have to produce messenger RNA, but you also have to downregulate the destruction of that RNA as well. And it turns out that what's happening is that you have these messenger RNA transcripts that are, in effect, part of the same family. They are regulated collectively up and down because they have the same regulatory sequences that are recognized by these enzyme complexes that break down RNA. And so what happens is that with regard to like pseudogenes and their corresponding coding brothers and sisters, is that because the pseudogene has sequence similarities to the gene that is encoded, when its messenger RNA transcript is produced, it actually functions as a decoy, where this messenger RNA degradation machinery will actually break down the decoy or the pseudogene RNA, allowing the coding RNA to actually be translated. And this also explains, again, the function of unitary pseudogenes as well, because they are part of these gene networks or these transcriptional networks. And they can, again, function as a decoy for genes that are part of the network that actually are coding, again, functional proteins. So we have this really elegant theory that explains, again, the functional role of pseudogenes, both unitary and duplicated, and also explains why these sequences look like like gene sequences. And so this is an argument for widespread functionality, to pseudogenes. Now, another criticism would be, well, this is in reference to those pseudogenes that are being transcribed, but for those that are untranscribed, there doesn't seem to be any kind of role. This doesn't apply to those. Well, the fact of the matter is that, again, as I mentioned, there's some very real concern that's being expressed now that maybe pseudogenes are actually being transcribed at a higher level than we thought, because of the similarity between pseudogenes and, again, those genes that would code for functional protein products. Because of the similarity in sequences, many times people are actually detecting pseudogenes transcripts, and they are ascribing them to the transcripts of their functional counterpart or their coding counterpart.
Well, that's a great, thorough answer, Dr. Rana, and it's so important because we know over the years this has been one of the evolutionists' favorite argument, and rightfully so. I mean, if these really are shared genetic mistakes and they're widespread and they're shared in the same genomic locations as chimpanzees, who they say are our closest cousin, that certainly would be something we should take seriously. And the genome-wide functionality that you're pointing out really is fascinating. So my next question then, Dr. Rana, would be, I have also heard this same thing with endogenous retroviruses. Even though we have discovered ERV function in cell stress responses, immune responses, embryological development, gene expression, I believe, they will still say, similar to the pseudogene argument, that there are only a few, and therefore the best explanation for the function would be co-option. Is this really true? Is this also a rare occurrence, like they say with the pseudogenes? And how to best respond?
Yeah, and I think that at this point, to be fair, the jury is still out, because we are in the stage where we are beginning to discover, again, function for endogenous retroviruses, and we're beginning to learn, what that function is and how these sequences are functioning in an anti-viral capacity. So we're still, again, in the very nascent stages of making sense of this. So I think it's premature for anybody to draw any kind of conclusions about really the prevalence of functionality. And there was a study done recently looking at, a particular class of endogenous retroviral sequences in the human genome. And it turns out that these sequences are functional, but they are only transcribed in the very early stages of embryonic development. And they're playing an antiviral role, protecting a very vulnerable embryo from viral invasion, but they also seem to be playing a regulatory role where they are activating certain genetic programs that are necessary for the early stages of embryonic development in the process of cell migration and in cell differentiation. But the point is that if you studied those sequences in cells from adults, you would probably conclude that these sequences were not functional because they don't seem to be active at that stage in development, right? Or once development is completed. And so we have to be careful about necessarily declaring something to be non-functional just because we are studying, cells at a particular stage of development or cells at a particular stage in the cell cycle. It could very well be that these sequences are functional only for a very limited time during the biology of the organism. And so we want to be cautious, I think, about declaring things. But to be fair, we're really at the beginning stages of recognizing function in ERV sequences. But this is a place where we would make a prediction, right? Is this an example of a rare occurrence of co-option, or is this a widespread phenomenon? If a design proponent or a creationist would say would predict that we should see extensive examples of function in EVEs and ERVs, whereas somebody that would advocate for the evolutionary paradigm would probably predict the opposite.
Amen. Amen. Well said. You nailed it when you said that it would be premature, because we would all agree that we are in the infancy of understanding the totality of the DNA language. And I think that's why us who hold to intelligent design and creation are confident enough to make these types of predictions. And I've seen you in your articles over and over again, Dr. Rana, make these predictions, functional related predictions when it comes to pseudogenes, ERVs, and so on and so forth. I wanted to ask you this, because this is another good article that you wrote. And I wanted to see if you could talk about the article's titled "Endogenous Retroviruses Protect Early Stage." No, actually, that's one we kind of just talked about. This one is koala endogenous retroviruses protect against retroviral infections. Can you talk a bit about this and how it applies to both the creation and evolution debate?
Yeah, well, again, the significant aspect of that article is that, again, there are these retroviral sequences in the koala genome that look as if they are actually able to disrupt the process of retroviral insertion, as part of the retroviral replication cycle. And retroviruses have RNA as their genetic material, and they are reverse transcribed into DNA, and that DNA can become incorporated into the genome. And but endogenous retroviruses, at least those that are transcribed, are mobile elements. They're retrotransposons, where they can actually move or make copies of themselves and insert throughout the genome. They can move around, and it turns out that mobility, that retrotranspositioning that ERV sequences in koalas can undergo, actually disrupts the incorporation of endogenous retroviral DNA into the genome. And so this is, again, identifying function for endogenous retroviral sequences, but also is explaining why they look the way they look. That the similarity to retroviral genetic material makes sense because of the nature of their functional mechanism. So it really ticks off a couple of boxes in terms of what is demanded of a creation model or an ID explanation, in terms of accounting for the presence of these materials in genomes.
Right. Another great response. Another great article. I got those mixed up because I've read so many of your articles in the last couple of days. Great articles, great information. And the question I would then have, Dr. Rana, based on what you're saying, and there's still so much we have to learn about genome functionality, including the possible functions of ERVs, is the appropriate question to be asking then is, are these really the ancient remnants of past viral infections, or are they created units of DNA function? And thus we predict function.
Yeah, well, again, from an evolutionary perspective, you would argue that these are the vestiges of ancient infections. That would be the interpretation, where again there was co-option in a process of functionalization. But until recently, the junk DNA argument, I think, really only made sense, or the presence of junk DNA in the genome really only makes sense in an evolutionary framework. And what we're seeing now is that somebody could legitimately view these sequences as part of the creator's handiwork. You could legitimately look at genomes as being, the work of an intelligent agent, as designed systems, because, again, we're making these kind of discoveries. So I think it's an important point to say that these discoveries aren't necessarily thoroughly undermining an evolutionary paradigm, but they're raising, I think, some sticky questions, for the evolutionary paradigm. But really, the significance of these discoveries is that it really makes it, I think, thoroughly legitimate now to view genomes as, as designed systems. And the interesting thing is that the more that we continue to learn about genomes, the more and more sophisticated these systems seemingly become. They're not becoming less sophisticated, but they're really beginning to become more sophisticated and more elegant systems.
Right. I mean, now we're finding out that there's multiple functions. For example, protein moonlighting, we're finding that the genome's multidimensional, it's nested. We have multiple overlapping codes. All these things, from my understanding, were not really expected 50 years ago. They weren't predicted 50 years ago. They had that paradigm where it was one gene, one protein, one function.
Yeah.
And we find Yeah, go ahead.
Well, and something else that I think is important to recognize is that, a lot of the sequences in the genome are actually playing a role in establishing the three-dimensional structure of the genome. This is something, as you're mentioning, Donnie, is a relatively new insight, that there's a three-dimensionality to the genome, and that three-dimensional structure is really critical in terms of the function of the genome, because it's bringing enhancer sequences that are at a vast distance from the genome that they regulate, or from the gene that they regulate. It's bringing them in close proximity to the gene. But there's other things that these so-called junk DNA sequences are doing. Some of the pseudogenes that don't appear to be transcribed are playing a role in chromosomal stability, for example. There's a proposal on the table that maybe some of the so-called junk DNA sequences are serving as a mutational buffer, that mutations accrue at a particular rate. And if you just assume that the mutations are taking place randomly, then you need a certain proportion of sequences that are buffer sequences that can absorb mutations without compromising functionality. And so that's a model that's being proposed. There's what's called a nucleus skeletal role, where it turns out that the ratio of the volume of the nucleus to the overall cell volume has to be tightly regulated. And if the cell gets larger, the nucleus has to get larger as well. And the way that you regulate nuclear volume is through the DNA content in the nucleus. And so there are some people that argue that there's a nuclear skeletal role that's being played by junk DNA sequences that are creating just simply enough volume in the DNA molecule to force the nucleus to be a particular size, which allows the support of the operations in a larger cell compared to a smaller cell. So these are, again, other functions that people have identified that look like they're valid functions, that also allow for the possibility that the functionality may be simply structural in nature. It doesn't have to be necessarily in terms of regulating gene activity or encoding, functional, information.
Right. So many good points there, Dr. Rana. That's amazing. Each one of those points you brought up could probably take an hour lecture just to kind of go over all the details. Here's something that comes to my mind as you mention all this, because I've heard the critics, and I'm curious as to your thoughts. I've heard them attempt to undermine this widespread functionality of the genome, specifically the human genome, because we've tested so much of it, by saying, well, we've done all these knockout tests, genetic knockout tests with mice, and we find that they still grow up, they have babies, they live their lives. Therefore, most of the genome really is non-functional. Have you heard that response? And what's a good response to that?
I have, and that represents an interesting challenge, to this idea of functionality. But it also could be that there is redundancy that's in the human genome or in the genome of organisms that is not being accounted for in these experiments. And so, if you think about systems that are well designed, many times there are redundancies that are built into those systems, particularly with respect to key, elements of the systems that are playing a critical or a vital role in functionality. And so, it could very well be that, that there's redundancy again in the genome, or it doesn't have to be redundancy in the sense that there's just simply duplicated function, but it could be that the redundancy is such that there's other mechanisms that would compensate for the loss of function. And so that I don't think is factored into these types of experiments necessarily.
Amen. Great response. And I've even read that many non-coding RNA genes, even pseudogenes that we spent some time on, are only expressed under certain conditions. So if some of these genes are being knocked out, and those genes being knocked out are not even expressed, or they're turned off because they haven't had the chance to turn on, maybe even through epigenetic means, then of course, the mouse is going to be fine at that point. So I better get to some of these questions too that are coming in from the audience, because I could ask you questions all day, Dr. Rana. You're a wealth of information. I appreciate that. So here's some good questions that have come in from the chat. Here's one from Logical, Plausible, Probable. It comes in the form of a super chat. So I appreciate that, John. John has interviewed you as well, Dr. Rana, on abiogenesis. I've linked that in the description box for people to check out. That was a great chat. So his question is, can you address the duplicate regions and how they are used based on cell type, efficiency versus waste / bad design? He says the usual Evo atheist talking point.
Gosh, I guess I'm not quite sure I get what Logical, Plausible, Probable is getting at. I'm really sorry. I'm not sure what's being asked there. I'm sorry. I think in some respects, we maybe touched upon that when we talked a bit about redundancy.
Right.
That redundancy isn't necessarily a waste, but rather it makes the system more robust. So if that's what Logical, Plausible, Probable is getting at, then I, but I'm not, again, I'm very, very sorry. I don't.
No, and that's, if John, if you can expand, because it could also mean too, because from my understanding, they'll look to neo-functionalization, where you get a duplicate gene, which remains, I guess, useless for a while, and then eventually it's subjected to a series of mutations that makes it functional. I'm not 100% certain either, so maybe we'll wait for him to address. But if it does have to do with that as well, we've talked about neo-functionalization. So I guess the next question has to do with, this question comes in from Doki Doki SFT Fan Club. And Doki asks, is the gene desert paradigm going extinct?
Yeah, I mean, yes and no, I guess. I mean, there are vast regions of the genome that don't contain, sequences that would code for functional protein products or functional RNA products. Again, like ribosomal RNA or transfer RNA, things like that. But what's being replaced with the gene desert is that these regions appear to be, again, functional, that they very well may be playing some kind of regulatory role. So I think the answer is yes and no, right? You can have gene deserts now, but the implication in the past was that those are just vast regions that were non-functional, and I don't think we can make that statement now. Something that this brings up is that a lot of times people are declaring regions of the genome to be non-functional based, again, on this expectation that flows out of the evolutionary paradigm. They've not actually gone in and done experiments that demonstrate that those regions are non-functional. And so a lot of this is an inference that's being drawn not based on experimental data, but really based on what the theory would entail. And so we want to be really careful, before we start making declarations, about whether or not parts of the genome are functional or not. There's a difference between, and even like with regard to the mechanisms for how so-called junk DNA sequences actually originate in the genome. These are mechanisms that are not unreasonable, but these mechanisms are really inferences that are being drawn based on the structural features that you look at, or based on some things that we know about the behavior of retroviruses and things like that. We've not actually necessarily directly observed these mechanisms in action generating, again, non-functional elements in the genome.
Amen. Well said. And it makes me think about the redundancy that you talked about earlier, and how redundancy is evidence for good design. It's evidence for forward thinking. But the evolutionist who holds to common descent, they go into it assuming that redundancy is bad design or evidence of evolutionary leftovers. For example, I think about the spare tire in the back of a car. To the evolutionist, that might be useless or wasteful. But if I enter an environment where I get a flat tire, it's a good thing I have that spare tire. It's redundant, it's necessary, and now it's going to help me out. It seems that a lot of the genome is indeed, as you pointed out, redundant for beneficial purposes. So I've got this from Logical, Plausible, Probable. So he says, kind of expanding on that question, Dr. Rana, he says the evolutionists claim you wouldn't have duplicate sequences if it was actually designed, avoiding the higher expression levels of specific genes based on cell type. So I'm not sure if that helps, but maybe they look to duplicate sequences as you pointed out, redundant. It's there. It's right. Not really doing anything.
Well, something else that I'm beginning to see too now is this idea that the complexity in the genome is unnecessary, right? So it's arguing that maybe there's functionality here, but that functionality is unduly complex. So this is another challenge that's being raised to the design argument that I think relates to the topic today. And again, redundancy would be something that would fall under that category. Well, to have redundant elements would be unnecessary, if you've got a functional sequence. But as you're pointing out, that redundancy, particularly if it's in reference to a critical system, and that redundancy doesn't necessarily have to be just simply duplicated sequences. It could be that the functionality represented by that region of the genome could be taken up by other aspects of the organism's biochemistry, but there's a redundancy in the system so that if some functionality is lost, there's a compensation that takes place, or again, a redundant element that could simply replace it. So, I think logical, plausible, probable is actually raising a really important point that again, we would see redundancy and duplication as essentially not a flawed system, but a robust system.
Amen. Well said again. I wonder, are you familiar with it? There's a paper, one of my favorite papers has to do with translational pausing and how a lot of these redundant elements, and I think specifically with third codon variation, third position codon variation, where I've heard a lot of critics say that they can use this because it's neutral variation, it's redundant, it's not really doing anything, and they can build phylogenetic trees, look at these nested patterns, and therefore conclude humans and chimps are related. But now we're finding out these redundant elements are not only there to correct error, but they're also involved in information flow. So we have multiple functions. And are you familiar with that, Dr. Rana?
I am, yeah. And for example, redundancy in the human or in the genetic code actually produces optimization that minimizes error that could or minimizes the loss of information that could occur as a result of both substitution and framework mutations. And so this is an instance that's related to your point, where redundancy in the system is actually producing an optimal system, and this idea of synonymous mutations.
Yes.
We're learning that these, where the mutation produces the same amino acid, we're learning that this isn't, again, these synonymous mutations are not actually synonymous, because different codons actually can control the rate of translation, as you're talking about, pausing at the ribosome. And that impacts the folding of the protein, which really produces a different type of functional protein, or a protein with modified function. So, this is really a profound point, because a lot of the theory related to molecular evolution is built upon the idea of synonymous and non-synonymous mutations. And if synonymous mutations really are not synonymous, that has catastrophic implications for molecular evolution.
Amen. So much great information, Dr. Rana. It's so true. That really is catastrophic, because there are specific evolutionary biologists that I've even engaged that thought, or that have pointed to this neutral variation, synonymous codons, and therefore the hierarchies that result from it. They have pretty well looked at that as just a absolute evidence for ancestry. And now it seems like, are we looking at neutral variation or functional variation? Yeah. And therefore, would you say these hierarchies are then there by design?
Yes, I would. And the thing is with nested hierarchies, again, where you have these sequences that are groups within groups within groups that presumably reflect an evolutionary history. But if you think about it, let's use the automobile as an example. Nested hierarchies are very much part of systems that are designed, particularly when those designs have a history where the design has undergone an evolution, if you will. And so, like, if you think about automobiles and the very first automobiles, clearly there are certain design elements there that all automobiles share, right? But then as we look at future generations of automobiles, there might be new design elements and new functionality that's introduced into the automobile, and that then becomes part of the standard automobile design. And so, future generations beyond that point, we're going to have both of those design features as part of as part of their design. And then later on, when there's a third design element that's introduced that then becomes mainstay, you now have three design elements that, if you looked at all the automobiles through that history of automobile design, you would actually group those automobiles within groups within groups within groups within a nested hierarchy. And so, nested hierarchies are found in human designs all the time. So it doesn't necessarily demand evolution as the only explanation. It's consistent with an evolutionary history, but it isn't exclusive to an evolutionary history. It could reflect the design history, which is what we're talking about, right? When we look at the history of life on Earth, it's a design history.
Well, that's a fantastic point, Dr. Rana, because I find it interesting that when we start from the Bible, we know that we're made in God's image. So therefore, there's something about us that reflects the divine. And yet we build, as you pointed out, we build systems in these nested hierarchical patterns. And yet we see nested patterns in the biological world as well. It's almost like we're getting an idea or a sense into how God designed based on the way we design.
Yeah.
We're made in his image.
Well, it's interesting to me because, so oftentimes the argument is that shared designs, whether the designs are in the form of a nested hierarchy or some other type of pattern, is evidence for an evolutionary history, right? And so you could also argue, well, it reflects common design, as opposed to common descent. And oftentimes I hear skeptics say, well, why would an infinite creator limit himself to just certain design features, right? Why would he limit himself to these archetypical designs that might be varied but still are archetypical designs? Wouldn't a creator who has limitless resources produce an incredible diversity of life on Earth, where all the different life forms look different? Well, the thing that's, I think, missing in that analysis is the fact that because of shared design features and because of these nested hierarchies, what we learn when we study one organism actually can be generalized and applied to a large number of organisms. And this allows us to study model organisms and from that develop general principles about biochemistry and biology. And so in a sense, the shared features that we see in biology reflect a design that makes biology even possible, as a scientific discipline. You could argue that that biology has been designed for discovery, designed to make it amenable to learn and make sense of it very readily and very easily. And so I see that as part of God's providential care. And so there's an elegant reason why a creator would create things with shared features and with nested hierarchies, because again, it lends itself to discoverability.
Amen. Well said, Dr. Ron. It makes me excited because it's a great time to be a creationist, to be an intelligent design proponent, because of the models and the possible predictions and discoveries, as you've pointed out with discovery. So great response. As we kind of wind down here, because time really has flown by with you and we're going on an hour and a half, I could certainly talk to you about these things all day, Dr. Rana. I want to get this one in. This is another super chat that comes in from Logical, Plausible, Probable. So again, brother, I appreciate the support. He says, some pseudogenes are shown to keep enzymes from reacting until they reach the correct destination. His question is, can you explain this and how it's a problem or might be a problem for common descent?
This is, I have not heard of this functionality for pseudogenes. I'm not, and so I can't really comment, but this I think exemplifies the challenge that's in front of us. It's becoming very hard to actually catalog the functionality for so-called junk DNA elements. I mean, it's almost like we're in an avalanche of discoveries that are taking place right now, identifying function for the junk DNA elements, or the so-called junk DNA elements, not just pseudogenes and ERVs, but other sequences like line and sign sequences. And so I can't comment on it. I'm going to have to. So thank you, Logical, Plausible, Probable, for pointing that out. I'm going to have to dig into this myself. But this is actually a good problem that it's really challenging to keep abreast of the discoveries that are identifying function. And again, this problem is something that is endemic to a ID or creation model approach. It's not something you would expect from within the evolutionary paradigm.
Amen. It's a good problem to have, that it's hard to keep up on all the incoming discoveries and data that ultimately supports our position and our position of design rather than descent. As we kind of wind down here, Dr. Rana, I was wondering if you could talk about an important class of RNA molecules called microRNAs, and you've written about them in a couple of the articles that I've read, and I think it's a fascinating topic.
Yeah, well, one of the things that we're discovering is that, there is, the so-called dark matter of the genome where you have all of these regions that are transcribed into what now is becoming like a zoo of RNA molecules that are of different sizes, that are playing regulatory roles. And microRNAs are in that category, just like long non-coding RNAs. And they all are playing, again, roles in gene regulation, and they're playing regulatory roles. And it, again, this is something that is, kind of a revolution, if you will, in molecular biology, is that there are all these RNA molecules that again are playing vital roles. And this goes again to the point that we were making very early on, is that you can't just simply assume that because something is transcribed that it's noise. Because these microRNAs and again the long non-coding RNAs would be included in the noise, at least with the original assessment of the ENCODE results. And again, we're discovering that these things are playing a vital role. So, we're in our infancy, and we're discovering things about the genome and its structure and function that are just highlighting the stark complexity, but yet it's also a sophisticated and elegant complexity that we're just beginning to grasp. And to me, it's just the genome is becoming an incredibly marvelous biochemical system.
Amen. And from my understanding, we're just beginning, Dr. Rana, to understand just how all the different windows and stages of the developmental process works, and how the coding and non-coding DNA portions of the genome interact, and in a way that coordinates how these structures are sequentially put into place to produce a fully functional organism. And therefore, it really is premature for the evolutionists to say that a lot of the genome is just spurious or junk or useless, because to me, and I wonder if you have these thoughts as well, a lot of the non-coding regions are probably involved in these various developmental windows.
Oh yeah, I think that's absolutely true. And it could very well be that, again, there are portions of the genome that are only active at certain points in development. And it's very difficult for us to assess that right now when it comes to the early stages of development, right? Because you're having to work in vitro on human embryo systems, and that opens up a whole can of worms, right? But the fact of the matter is, it's very difficult at this point to truly get an assessment. But I suspect that very much is the case, and I think one of the things that is going to be something I predict happens as we move into the future, and to some degree it already is that the regulation that's taking place in the human genome, and again the genome of other organisms, is so sophisticated and complex that it's really going to take a systems engineering approach to really figure out what's going on in terms of regulation. And I think we also are going to discover that not only is systems engineering critical to interpret the regulatory networks that are involved in regulating, the activity of the human genome, that I suspect we're going to discover regulatory systems that inspire new advances in systems engineering as well. That we're going to learn things from the human genome that are going to open up new vistas in that area of engineering. So that's another prediction that I think we could make, that I'm not sure an evolutionary biologist could make the comparable prediction.
Amen. Well said. And I would agree that is a good prediction, and where we are heading, that's probably going to be confirmed. So I'm going to end it with this last question. This is a question that kind of came in all the way at the beginning to respect redefined living. I want to get it in there. Hour and a half really has flown by. So much great information, Dr. Rana. So the question is, when you say information, is that analogous or literal when concerning the genome?
I think the information is literal information. The information in DNA is digital information, there's discrete units that are reflected in the nucleotide sequences, and when we're talking about a gene, really in the codons, the coding triplets. But in addition to that, we're now learning, as we talked about, that there's a three-dimensional structure, to the genome, and that three-dimensional structure could be viewed as being analog information. So there's really both digital and analog information embedded within the genome, where digital information is discrete and analog information is continuous. And so when you talk about three-dimensional shapes, there is a continuum that those shapes are on, and so those shapes are actually have information instantiated in them, and so I think it's appropriate to think of the genome as having both digital and analog information. So it's an incredibly sophisticated system in that respect. And again, we're just beginning to understand how to decode that information. So, what's remarkable is that embedded in a sequence of DNA is not just simply, the genetic code, which defines the information, but there is a histone binding code. There is a transcription factor binding code. There is a spliceosome code that are all overlapped and stacked on top of each other in the same sequence. So you have multiple overlapping codes that are taking the same sequence of nucleotides and communicating different sets of information to different aspects of the cell's machinery. That to me is mind-blowing. And in fact, the genetic code that we see in nature, which is in effect universal, is redundant, and that redundancy allows for multiple overlapping codes. And the genetic code in nature seems to be optimized among random codes that could be imagined to harbor multiple overlapping codes. So, the information that's embedded in DNA is so complex and so sophisticated and so elegant. I think an information scientist would struggle to produce a system that could harbor multiple overlapping codes at the same time.
Wow, great response. It really is science fiction what we're seeing on the molecular level. It reminds me, what you said reminds me of Michael Behe's book Darwin Devolves, with how sophisticated and complicated the genome is, and yet evolutionists want to say that mutations, which I understand are mostly damaging and functionally compromising, could take a single-celled like ancestor into, let's say, a whale over time. When with such a system that's so complicated, it's more likely that mutations are going to damage or degrade functionality rather than improve it or make something better.
Yes, because that's true. I mean, when you have highly optimized systems, any kind of change to that system is going to move you off of that optima, right? And so I think that's very much the case. But what's also remarkable is that mutations are a consequence of the fact that we live in a world that conforms to the laws of thermodynamics. And yet what we see, in the cell is all these mechanisms that are put in place that actually turn what could otherwise be deleterious mutations into neutral mutations or mutations of minimal consequence. And so there are all these redundancies that actually create robustness against mutations. And this is in the nature of the genetic code itself. It's within the fact that there may be sequences that are serving as mutational buffers. It's in the redundancy that we see within the system. And so plus, not to mention DNA repair mechanisms that are in operation as well, which is in and of itself a whole nother facet of the design argument. And so, so the remarkable thing is how few mutations actually compromise function. In light of the fact that these systems that we're looking at are so highly optimized.
Well, like you said, we've got these DNA repair enzymes and systems that help repair error. And if we didn't have those, we'd probably be extinct in a generation. I mean, that's thinking ahead. That's forward thinking, which points us back to a forward thinker. And the buffering makes me think of your car, the treading on your tires. You know, over time that slowly wears down, but it does take time, and it's there to prevent the the ruining, I guess, of your tires after one drive. You got a lot of buffer there that's designed that way, just like the genetic system. So great points, Dr. Rana. I want to hand it to you. We're going on an hour and 40 minutes. We still got a great chat. So much great information. We touched on so many good topics: pseudogenes, hierarchies, endogenous retroviruses, junk DNA, gene deserts. So a great, thorough show. I want to thank you so much for giving us your time, for giving me your time for this important program. I know you're busy, Dr. Rana. So I wanted to hand it to you for some final words, final thoughts before we close it down.
Yeah, to me, I think we're at an exceptional time in scientific history when it comes to genomics, and it's remarkable to me to think that it was about 20 years ago that the first draft of the human genome sequence was produced, and what we've learned in the contravening 20 years has been nothing short of absolutely astounding. Particularly for people that hold to an ID perspective or a creation model perspective. I suspect that the next couple of decades are going to unleash even more profound discoveries. But to me, the thing that's so encouraging is that the more that we're learning about the human genome and the genomes of other organisms, the more that we're seeing a case for design, that is a case for design that is mounting, and I think there's going to be a point in time when we look back and we could never imagine anybody thinking that the genomes of organisms were anything other than these incredibly elegantly designed systems that suggest the work of a mind.
Amen. Well said. Well said. People are in the chat are saying thank you for a great presentation. John did list the book that he was recommending to you, but the chat's moving too fast, and unfortunately I lost it. I apologize. John, maybe you can email it to me and I'll email it to Dr. Rana. We've had a great chat, great final words, Dr. Rana, and I can't thank you enough. So God bless you. God bless everybody in the chat. Please share this around. This content is important, and we'll see you guys later. God bless. Standing for Truth is out.